All right. So today is probably what you've all been waiting for two whole semesters now, and that's the heart. Okay. Now, last time we talked about blood and that the whole goal of blood, right, is going to be to carry nutrients to our tissues, and then carry waste and stuff away, right? Well, the heart, then, its job is simply going to be to supply the force necessary to move that blood through the vessels. That's really it. Okay. That is our whole goal, is supplying enough or pushing that liquid with enough force that we can get it to go out to all of the tissues adequately. So to get enough blood going to all of the tissues that all of those cells and stuff can get everything they need. Now we call this perfusion, this rate of blood that gets delivered per unit time. Okay. So delivery of blood per unit time is going to be what we call perfusion. Now, we normally measure this in milliliters per minute, okay, per gram of tissue, okay? We need to make sure that we have enough. If we do not have enough perfusion, this simply means that some of our tissues are not going to be getting enough nutrients. They're not going to be getting enough oxygen. They're not going to be getting enough sugar. They're not going to be getting enough fat, and so on and so on and so on, okay? Which is problematic. That also means that they're going to have buildup of waste and all that kind of fun stuff. So we definitely don't want that. Now, we have mentioned a couple times that we have different types of blood vessels. We have arteries, veins, and capillaries. Arteries, importantly, carry blood away from the heart. Okay. They do not necessarily carry oxygenated blood. Most do, but pulmonary arteries do not. Okay. So arteries are going to carry high pressure blood away from the heart. High pressure because it is going to be close to the heart. The heart will have just squeezed and thus pushed that blood. Okay. Veins are going to carry blood back to the heart. Okay. Systemic veins will carry deoxygenated blood, but pulmonary veins are going to carry oxygenated blood to the heart so that then it can get pumped out through the systemic arteries. Okay. Now, capillaries are going to be our exchange site. Okay. Whenever we need to do gas exchange or nutrient exchange or whatever that may be, we're going to do that through or in our capillaries. Whether that is between the blood and air, say in our lungs, or between the blood and our tissues. Okay. So here we have a kind of general layout of the cardiovascular system. We can see all of the systemic arteries and veins and here, but we can also see this set of arteries and veins right here, kind of where our lungs would be. So this would be our pulmonary arteries and veins around here. Okay. Now, our capillary beds, like I said before, these are going to be found kind of throughout all of our tissues. But we have our pulmonary arteries, or pulmonary capillaries, is where we're going to have gas exchange between the air and the blood. So this is where we're going to oxygenate the blood, and then we're going to kind of pass off any CO2 from the blood outside. And the opposite of that's going to be true in our systemic capillaries. Now, our heart has kind of two main sides, and it has four main chambers. We have a right side of the heart, and we have a left side of the heart. The right side of the heart is going to carry or deal with deoxygenated blood. Okay. So the right side receives deoxygenated blood and then pumps it towards the lungs. That is the goal of the right-hand side of the heart. The left-hand side of the heart is going to receive oxygenated blood from the lungs and then pump it to the rest of the body. Okay. So the right side is going to be where we're going to get blood from the body that has been deoxygenated. It's going to then pump it to the lungs where then it gets oxygenated, and the left-hand side will then receive that oxygenated blood and then pump it out to the rest of the body. Now, these two sides, each of these are going to be split into an upper chamber and a lower chamber, okay? So these superior chambers are going to be what we call atria, okay? So the upper chambers are atria, the lower chambers are ventricles. The upper chambers are atria, the lower chambers are ventricles. Now, the upper chambers are going to be the parts of the heart where we're going to be receiving the blood. Okay. So blood will enter the heart in those atria. So blood from our superior and inferior vena cava will enter our right atrium right here, and then flow down into our right ventricle. Our right atrium can then squeeze and make sure that right ventricle is super filled, once again with deoxygenated blood. So then when that ventricle contracts, it will push that blood up out into the lungs. The left side, basically the same thing. Left side is going to receive blood from the lungs, make sure that we can fill up this left side ventricle, and then pump it out through the aorta to the rest of the body. Okay, so right side deals with deoxygenated blood, left side will deal with oxygenated blood. We've also got what we call the great vessels. Now, the great vessels are the primary the primary blood entry and exit points from the heart. Okay, so for our deoxygenated side, we have our veins. We have the superior and inferior vena cava. These are going to contain all of the venous return, so all of the blood return from the body. Okay, so whether that is blood from your head, your neck, your arms, your chest, all of that stuff is going to come into our superior vena cava. Blood in the inferior vena cava is going to come from anywhere basically south of the heart. Okay, so anywhere from your abdominal cavity, your legs, anything like that, all of that blood will get routed back to the heart through our inferior vena cava. But both of those will have entry points into our right atrium. Okay, so the superior and inferior vena cava will drain into our right atrium. Now, if we look at the other deoxygenated vessel here, we can see this big T-shaped structure here. This is our pulmonary trunk. Okay, it's T-shaped because it's going to split. We're going to send some over here to the left, and we're going to send some over here to the right. But this vessel is an artery that's going to be carrying blood away. In this case, it's going to be carrying deoxygenated blood towards the lungs. Okay, but it's going to be sent out through the right ventricle. Okay, so superior and inferior vena cava drain into the right atrium. Right atrium drains into the right ventricle. Right ventricle then pushes blood out through the pulmonary trunk. So that is all dealing with deoxygenated blood and dealing with the right side of the heart. The left side of the heart, then we'll have our pulmonary veins. So our little red pulmonary veins here, so superior and inferior, both left and right, will drain into our left atrium, which then gets pushed down into the left ventricle, which then gets pushed out through the aorta. So the red vessels here are the great vessels for the left side of the heart because we're now dealing with oxygenated blood. Okay, so pulmonary veins are going to return oxygenated blood to the left side of the heart, so then we can pump it out through the aorta to the rest of the body. Now, which of these do you think is going to be dealing with higher amounts of pressure? The pulmonary trunk or the aorta? Keeping in mind that your lungs are here, the heart is here, but so your lungs are this close, but I need to be able to pump blood all the way down to the ends of my toes or the ends of my fingers from the left side. So which of these is going to be dealing with higher amounts of pressure, the aorta or the pulmonary trunk? The aorta, by a lot. If you look at the walls of the aorta, it is composed of lots and lots and lots of elastic connective tissue, specifically so that it can stretch. Every time your heart beats, it can then receive that energy wave as the heart is contracted really, really hard. It can then receive all of that amount of blood and not kind of rupture or split, usually. Sometimes, though, people's aortas do split, or at least the walls will start and try and split and things like that. We call that a dissection, where you essentially have a tear in the muscular wall. As long as it doesn't open up all the way, you can sometimes survive it. Now, separating the upper and lower chambers and the great vessels, or particularly the exit great vessels, so your pulmonary trunk and aorta, separating the chambers and the pulmonary trunk and aorta from the heart are going to be valves. Okay, these valves are going to be very, very important. These valves are, well, what a valve is. These are basically going to be one-way paths through which the blood can pass, but it can only move in one direction. Okay. Now, these are going to be made out of some really dense connective tissues so that they are incredibly strong and also very, very resilient. They're also going to be somewhat hard. Okay. This basically is going to allow them to act kind of like a door. Okay. But the idea here is that by having these little flaps, if say we're trying to move blood from our right atrium into our right ventricle, the right atrium can contract and push with a little bit of force on some blood, and it's going to cause these little doors to open. So imagine like this door here. If I want to walk out this door, it's fairly simple. I just push on it and it will move that way. However, there's an edge to this door that prevents it from being able to come the opposite direction, right? Like I can pull on this thing all I want and I will never be able to pull it back inside, okay? The valves in your heart aren't quite as one way as that, but they try to be, okay? We have some little cords attached to these valves or to the flaps of these valves to prevent them from being able to move the opposite direction. That's very important because the atria are going to push with a little bit of force on these valves, but the ventricles are going to push with a lot, okay? Because they have to push things out all the way to the body or all the way to the lungs, so they have to push with a little bit more force than the atria do. So we need to make sure that these doors don't slam themselves open the other direction. So they're going to be held with some little cords that we call chordae tendineae here to keep them from moving backwards, okay? But the whole idea here is that we try and keep the blood moving only one direction. We don't want it fighting against itself because that's going to cause turbulence, and that's also going to be a massive waste of energy because that means we're going to have to pump blood more than once just to get it to move through the heart. We don't want that. We're trying to get the blood to move through in as linear a fashion as we possibly can. So having to pump the same red blood cells multiple times within a single kind of couple of seconds would be problematic. So we prevent that backflow using these valves, okay? So we're going to have valves here, like I said, between the upper and lower chambers. We're going to call these atrioventricular valves because they are between the atria and the ventricles, thus atrioventricular. And we've also got our semilunar valves in between our pulmonary trunk and the right ventricle and the left ventricle and the aorta. The one between the aorta and the left ventricle, we're going to call the aortic semilunar valve. The one between the pulmonary trunk and the right ventricle, we're going to call the pulmonary semilunar valve. They're called semilunar because they're basically shaped like half moons. Okay, so if you look at their picture, the little flap of connective tissue looks like it's roughly the shape of a half of a moon. Thus, we're going to call it a semilunar valve. Okay, now the idea behind this shape is actually fairly simple, though. Namely, that we have an opening right here between the two flaps. So if we push with some force, the blood can, say we push this way, it can actually force these flaps to kind of fold out and allow the blood to pass through, and everything is fine. However, when we remove the force, well, that means we have blood up here, right? And we've got gravity pulling that blood back down, right? Because that gravity is pulling the blood back down, it's actually going to pull these flaps closed on themselves. So these two little flaps will meet each other right here in the center, and all of a sudden they can't move anymore because they're basically meeting. So these cups now are basically going to now fill with blood as they are waiting for the next beat of the heart to push that blood past. Does that make sense? So because we have this kind of cup shape, they're going to collect a little bit of blood, and it's going to prevent that blood from being able to flow back into the chamber of the heart where we have to spend energy to push it back out again. Once again, we're trying to prevent waste. We don't want to waste energy in this process at all. So those are some of the main structures of the heart. We have the two sides of the heart, where the right side deals with oxygenated or deoxygenated blood. So the right side, does that deal with oxygenated or deoxygenated blood? Yeah. Deoxygenated blood, okay? The left side, by comparison, is going to deal with oxygenated blood. We have two chambers on each side. Which ones are the upper chambers? Atria. What are the lower chambers? ventricles. Okay. Then we've got our great vessels. Which of the great vessels is going to carry oxygenated blood towards the tissues? The aorta. Which of the great vessels are going to carry deoxygenated blood to the heart? Somebody else. Perfect. Superior and inferior vena cava. Fantastic. Okay. So that is kind of our main goal then. So we should already see how this whole process is going to work, right? We have four chambers. Upper and lower. So we have atria, ventricles. I have them drawn in color where we're dealing with oxygen. Red is oxygenated, blue is deoxygenated. All we're going to do then is we're going to take blood and enter here by our superior and inferior vena cava. That blood then passes through this valve, okay, which is on the right side of the heart. So this is our right atrioventricular valve. Then we pump out through the pulmonary trunk. Then we pump back in via our pulmonary veins, down through our left atrioventricular valve, and then out through the aorta to the body. And we're going to do this 70 times a minute. Over and over and over and over again. But that's at rest. If you go faster, it just speeds up. That's simple. When we need more blood, it pumps faster, but it also pumps harder, because we will fill these chambers with even more blood so that we can then press with more force and push more blood with more force faster whenever we need to speed up the blood rates. Okay. But this is the basic pathway that our blood is going to take through our heart. Okay. So our basic pattern is right side of the heart to the lungs, to the left side of the heart, then to the tissues, and then back to the right side of the heart. Over and over and over and over and over and over and over again until you die. Some people think that you can only do this process so many times before you die, so they don't exercise. That's actually not a joke. Some people literally think that your heart can only beat so many times. Oh, well. All right. Now, there are different, wait, what is happening? I've never seen, what is over here? I don't know what this is. So I'm going to click close and hope it's okay. Sure. All right. If you've ever heard of someone having edema, this is basically just going to be swelling. We can have swelling occur because we have some sort of blockage in the system, or at the very least that we have the inability to get blood out of a tissue. Okay, so if you've ever seen someone with swollen legs, for instance, that's a pretty common one, especially for folks that are diabetic or pre-diabetic or something like that. They'll oftentimes exhibit some edema in their legs. It's basically just swelling because blood will pool in their legs because their legs are nice and low away from the heart. So they're not getting gravity's help to bring that blood back. So they will have some of that blood pool down there. And that can cause a little bit of a problem. But that blood pooling is just simply going to be, like I said, because we're having an issue getting the blood to return. This could be an issue with us not getting enough pressure out of the heart. Okay. Now, the heart itself is going to be found between our lungs, behind our sternum, and slightly to the left. Okay. It's going to be found in its own little cavity. So whenever we learned about the different body cavities, you probably remember that we talked about the thoracic cavity and the abdominal cavity. Well, Inside the thoracic cavity, there is another cavity called the mediastinum. Okay. The heart lives in the mediastinum, as do the great vessels. Okay. So the mediastinum is basically going to be filled with the heart and the great vessels. It's going to be this small space in between the lungs. Okay. Now, the top of the heart is going to be found roughly even with the top of our second rib. Okay. So if you count from your first rib down to your second rib, that's basically going to tell you where the heart is going to start. The apex of the heart is going to be found down here at the top of rib pair 7. Okay, or sorry, rib pair 6. Okay. So that's basically going to kind of give you the size of your heart. As you can imagine, someone that is larger is likely going to have a larger heart by comparison. Okay. Now, one fun thing that's happening with our heart too, though, is that we're also shifted slightly to the left. Okay, that's why if you've ever had your doctor listen to your heart sounds, they're going to spend more time on the left-hand side of your chest. Okay, and that's simply going to be because the left-hand side of the heart kind of pokes its way out over here on the left-hand side of your sternum a little bit more. Okay, where the apex of our heart is actually found well over here on the left-hand side. So we are not centered. This also means, though, that we have to have a space in our left lung where our heart can live. So if you were to actually look at the lungs, the left lung will actually have a notch out of it that we actually call the cardiac notch, which is basically the space inside which the heart is going to live. So here we can see kind of part of that mediastinum. We can also see the left ventricle here, or the left side of the heart, and all that fun stuff in here in this particular cross section. Now, the heart is going to be surrounded by a couple layers of connective tissue called the pericardium. Okay, the pericardium is going to be composed of So the pericardium is going to be composed of two primary layers. We have the fibrous and the parietal layer of the serous pericardium. These are going to make up what we call the pericardial sac. This is quite literally the bag that your heart lives in. However, this bag is hard, okay, or at least firm. It's not soft like a baggie where it's super malleable. We don't want it to be super malleable. Why? Because we breathe. Okay. If this bag was too malleable, every time you took a deep breath, your lungs would be able to change the shape of that baggie and your heart would not be able to fill with blood, which would be problematic at best, against the point at worst. Okay. So we have to have the pericardial sac be fairly firm. Otherwise, the lungs would be able to encroach too much. So we have to make sure that we have space for the heart to expand inside. So the outer layer of our pericardial sac is our fibrous pericardium, and it is, like I said, fairly tough. Okay. On the inside of that, we're going to have a little layer of so the parietal layer of our serous pericardium is going to be found on the outside of that. So we have this nice little space here. And then finally, we have our visceral layer of our serous pericardium actually touching the surface of the heart. So this should sound at least somewhat familiar to like the brain, where we had that inner layer that was actually touching the surface of the brain. So here we have this inner layer that is touching the surface of the heart. Okay, so that is our visceral layer of our serous pericardium. But we do actually have a little bit of fluid in this space in between as well. Okay, that fluid in between our visceral and parietal layers is very, very important. Because it's going to make sure that we do not have grinding between those two layers so that we do not damage either of the layers as the heart is expanding and beating and things like that, because that's going to be a little bit of friction every time that occurs. We want to make sure that we have a little bit of fluid there so that we can prevent that friction rub. We do not want that. If that happens, you can hear it. You're going to hear this as a crackling sound. Like anytime someone's heart beats, you'll actually hear this as like a little bit of a crackle because of the roughened portions of that visceral and parietal layers touching each other without that fluid. So we definitely do not want that. However, we also don't want to fill this space with fluid because fluid doesn't compress, and every time our heart would try to fill with blood, it wouldn't be able to. So we wouldn't be able to get enough blood into the heart. That would also be a problem. This can sometimes happen if people get particular types of bacterial infections or fungal infections and stuff like that. As you all can tell, there's lots of stuff that can go wrong in all of these processes. All right, so we've got our chambers. We have the right and left sides of the heart. We haven't talked about something, though, that is incredibly important. When I said the aorta, where does the aorta carry blood? It carries blood everywhere except the heart. This is all muscle. If I need to pick up something with some muscles, how do I do that? How do I generate that force? I have to send electrical signals and stuff to it, right? In that process, I'm also going to burn through ATP, right? Which means I have to get oxygen to those tissues at some point so that I can make up for the ATP that I burned and continue to make some more, right? Which means we have to have an oxygen supply for the heart muscle. That oxygen supply for the heart muscle is going to come from these blood vessels here. Okay, these are what we call our coronary vessels. Okay, we're going to have some that we're going to feed the right-hand side. We're going to have some that are going to feed the left-hand side. Okay, so as we split our blood into our aorta, right after our aorta kind of exits the heart and it goes through that aortic semilunar valve, there's going to be two little offshoots. Okay, these two little offshoots are going to be our coronary arteries. The coronary arteries are going to be what are going to supply the heart with its blood. Okay, so even before we enter the ascending aorta, we're going to have the coronary arteries split off. Okay, these coronary arteries are going to fit in what we call the coronary sulcuses. So if you look at the outside of our heart here, so here we're looking at the outside of the heart. This big structure right here is our right ventricle. So here is our left ventricle here. The right and left atria are going to be covered in these little ear-shaped flaps that we call auricles, okay? But this little groove in between this flap and the left ventricle is a groove. So just like a groove in the brain was called a sulcus, the groove here in the heart is also called a sulcus. In this case, it's the coronary sulcus. So this is where our right coronary artery is going to live. The right coronary artery is then going to supply all of the right atrium and ventricle with all of the blood that it needs in order to beat and do its job. Notice that we have a whole lot more blood supply going down to the ventricle than we do into the atrium. That's because the ventricles are going to press with a whole lot more force than will the atria. The atria are basically what we call a pass-through sort of chamber. Yes, they can squeeze so that they can make sure that they're completely empty before the ventricles contract, but they're not having to squeeze with as much force because they're basically pushing blood a couple centimeters. That's it. The ventricles are having to contract with enough force to push blood up through arteries and then in through capillaries and back. That's how much force these have to exert. So the ventricles are going to contract a lot more force and thus need a lot more blood. So you're going to see a lot more blood supply going down here towards the ventricles than you will into the atria. Okay. Now, as we get towards the bottom of the heart, one of the big branches that we're going to see off of our coronary artery here is going to be what we call the marginal artery. It's called the marginal artery because it's basically down here at the base of the heart. Okay. Because the heart is somewhat turned and kind of turned backwards. So it's kind of tilted towards the back. So the apex is pointing posteriorly. It's going to kind of sit on the bottom of our right ventricle. Okay. So this is still our right ventricle where most of the left ventricle is going to be found posterior. Okay. So we're kind of sitting on this right-hand margin. So thus we're going to call this the right marginal artery. Now, we have this other big blood vessel right here wrapping around the left-hand side. So this would be our left coronary artery. One of the big splits or one of the big branches off of this one, we're going to call the anterior interventricular artery. Sounds like a lot of words. Sounds like it might be difficult to learn that one, except it tells you exactly where it is. Anterior tells us that it's on the front of the heart. Interventricular tells us it's between the what's? Between the ventricles. And artery tells us it's going to be the red one. Okay, so the anterior interventricular artery is going to be found right here, and that's what's going to allow us to see the separation between our right and left ventricles, where this kind of big point right here is the apex of the heart. Okay, so like I said, we have these big flappy structures here that we're going to call auricles. They look like little ears, and these are going to be found on our right and left atria. Now, when we look in the back, things look a little bit wonky, okay? Namely that we don't see those arteries quite as well. We can still see the circumflex artery a little bit. Now we can see our right artery a little bit more. We also have a big artery right here separating our right and left ventricles. So this would be our posterior interventricular artery. But we can also see where all of the veins on the heart are draining back as well. So all of the veins of the heart are going to be draining their fluids back towards this structure right here. This is our coronary sinus. So the coronary sinus is basically going to be the collection port or the main vein for our heart. So after the blood that gets pumped out to the ventricles and out to the atria is deoxygenated by those cardiac muscle tissues, it then flows back towards our coronary sinus so that it can empty back into the right atrium, and then start the process all over again. Okay, so we have exits immediately at the end of the heart or before the aorta really even starts, and then we are able to re-enter back into the heart. So this way the blood that's feeding the heart isn't having to go very far. We're able to run into this kind of fairly short circle before we get sent back out. Okay. We don't have to worry about entering the superior inferior vena cava or anything like that. It goes right here into this coronary sinus. Now, if you have ever heard of someone having a heart attack is going to be a blockage in any of these arteries, whether it's in our right or left coronary artery, or in, say, our interventricular arteries or the marginal arteries. Any total blockage in any of those will cause a heart attack. Okay, now, when that happens, that means that we stop getting blood flow to some amount of the muscle. If it happens in one of these coronary arteries up here, so before they have split, all of a sudden we can have large sections of the heart not getting blood flow. Even for a few moments, this can be enough to have someone pass. Which is why a blockage in, say, our right or left coronary arteries, or these right and left coronary arteries, are sometimes called the widowmakers. Because one of them if you have a blockage here, and that's what causes your heart attack, it may be irrecoverable. Just simply because it's going to shut blood off before that blood has actually had a chance to split off. Like, say you block right here, well, this whole right side of the heart's not getting any blood anytime soon. That's a problem. That's a problem like now. Like, oh, that happened, you might be dead before the ambulance gets here. If that happens out here, you might be okay. But if it happens here, yeah, it's definitely bad. Now, we can also see from this posterior side, this is going to be our right atrium. We can also see where our pulmonary veins are going to enter back into the right atrium. I think just the spot just looks weird. Like a squid or something. All right. Now, the layers of the heart wall are also going to be quite different. Once again, our lungs are 2 or 3 inches away from your heart at their furthest. Your toes are 3 feet away. I just realized that's my hand. That's not my toes. It's basically the same. Long way away from the heart. Okay. Because of that, the left side of the heart is going to be beating much harder. So the aorta is going to be under much higher pressures. But this also means that the left-hand side of so the left ventricles are going to be massive compared to the right-hand side. Why? Because we have to have more muscle so that we can exert more force on that same volume of blood, because we have to push it that much further. The right just pumps to the lungs, which are very nearby. The left has to exert a high enough pressure that we can get it everywhere through systemic circulation. Now, the heart walls themselves have three main layers. We talked about the epicardium a little bit. The epicardium is going to be covered in this layer of simple squamous epithelium. Then we're going to have a thin layer of fat. Underneath that, we're going to have a bunch of muscle. The muscle here is going to be cardiac muscle. Now, remember that cardiac muscle has branches. So we have these kind of Y-shaped cells. And then we have these little in-between sections that we call intercalated discs. So where two cardiac muscle cells touch, we have an intercalated disc. That intercalated disc is very important because we can actually flow ions and things like action potentials through the margins of separate cells. When one cell sends out an action potential or has gone through its action potential that, remember, gets propagated down the length of the entire cell, it can then propagate that signal to the next cells that are touching it. This way, we can have the entire heart, which is basically just a big network of these cardiac muscle cells, contract in a very particular order, just based off of how these little Ys are connected to themselves. Because this one, when this cell contracts, it's going to cause this cell, or these cells past it, to then contract, and then these cells past it to contract. So we can actually send signals through the heart muscles themselves, not with nerves, but through actual cardiac muscle cells, which is cool as shit. Okay, now on the inside of the heart, we have our endocardium. Okay, so the endocardium is another layer of simple squamous epithelia on the inside, so another layer of endothelium. This is basically the same thing that we're going to find in our blood vessels. All right, now we can go ahead and look at some of the other kind of minor features, or at least smaller features. First off, we have our fossa ovalis here. This is basically going to be the entrance for our coronary sinus, near our opening for our coronary sinus. So these are kind of these areas right here. This is where blood is going to be entering the right-hand side of the heart. We can see these little cords attached to our atrioventricular valves. We call these the chordae tendineae. That's just a fun name. I like all the Latin stuff, and this is about as Latin as something can be, okay? These are also going to be connected to these little bits of muscle down here, these little pillars of muscle, which is fun. We're going to call these the papillary muscles. These are basically here to open and close open the valves. So say we need to open this valve so that we can allow blood to flow from the right atrium into the right ventricle. Then these papillary muscles can contract and open the valves to make sure that blood can pass through and drain into the right ventricles. And the same thing is going to happen over here on this side as well. Your heart, like I've mentioned it a few times, has to be able to expand, right? It's filling with blood. Whenever it fills with blood, that's going to change its overall size just for that moment. But every time that we have an organ that needs to be able to fill and then contract, we have to have some ability for the inside of that organ to be able to kind of round itself out and then maintain structure and stuff when it contracts. That extra volume and those extra ridges that we're going to get in the heart are these guys back here, okay? This is what we're going to call the trabeculae carnae. That's another one that's just super fun to say. Okay, so the little lines that you're going to see kind of making up the insides of the chambers of the heart, we're going to call these the trabeculae carnae. These, though, like I said, are basically going to allow these chambers to expand, okay? Whenever the heart is fully expanded and these ventricles are completely filled with blood, you really won't be able to see the trabeculae carnae because they will be stretched, okay? Like, so say you're talking about like in the midst of strenuous physical activity, so your heart is filling itself as much as it possibly can in between beats. When it is completely filled with blood, these things are basically going to be stretched out as far as they can, so you wouldn't be able to see them as well. But when the heart is contracted, like it is in this case, somewhat contracted like it is right here, so you can see a little bit of these folds, basically showing you, hey, we have more stretch to give. Oh, one fun thing with these atrioventricular valves, like I said, the whole idea here is that we're preventing backflow of blood. We don't want to have to pump blood twice. We have one on the right-hand side that we're going to, oftentimes, you'll hear it called the bicuspid valve. That's because it's basically only got two little cusps. The one on the left will sometimes be called the tricuspid valve because it's got three. But these valves can actually be replaced. One bad thing that can happen to folks over time is that sometimes these valves can start to leak. Something happens, those cords break because they had something like, say, strep throat and never got it fixed. Those bacteria can work their way down into your heart, and then they'll literally eat away at those chordae tendineae, and then one day those chordae tendineae can pop. So when those chordae tendineae break, that means all of a sudden your valves can move backwards when your heart goes to contract, which is a problem because once again, that's backflow of blood. That means we're going to have to try harder just to pump blood around. The last thing we want to be trying hard to do is pump blood. Okay. However, you can fix these. They can fix these using human valves from people that have passed away. They can fix these with horse, valves from horses, valves from cows, valves from pigs. But they can also do it with synthetic valves and stuff like that now too. So there's lots of ways that these things can be repaired. So they would literally cut your heart open and put in a new one, which is fun. It's crazy some of the things that people can do. So here you can see the valves. We can see our left atrioventricular valve here, right atrioventricular valve. So 1, 2, 3. So thus tricuspid, 1, 2, thus bicuspid. And this picture is also showing you kind of how the blood flow can force these things open, but then the blood pooling is going to cause them to close. It's kind of like what I was trying to draw you up here. And then the opposite of this is basically going to be true for our atrioventricular valves. When the heart contracts really, really hard, that's going to basically slam the doors on those valves. So much so that whenever you actually listen to someone's heart sounds, you're not hearing the muscle beating. That lub dub that you're hearing is actually the sound of these valves slamming shut. And that's a big part of that is because like muscle is fairly soft. So you're not actually going to be hearing the sounds from that just because it's soft. They're not really making any impact noises when they hit. But this is going to be dense connective tissue, so you can actually hear it. You can also hear it when one of your friends bites into one of these. So one of the worst things I've ever done to another human being is I convinced one of my buddies to eat some raw dove hearts one day when we were hunting. We were cleaning a whole bunch of dove hearts after we'd gone hunting, or a whole bunch of dove, and he was taking forever. Like there were 3 of us that hunted, and he was taking 3 or 4 times longer than me and my other buddy, like cleaning ours. We were like, dude, what are you doing? And we look over and he's got like a little pile of hearts, like sitting next to him. And it's like, why? You're just why? And he tried giving us some like spirit of the animal nonsense, like basically saying that this is like what the Native Americans and stuff did. Like whenever they harvested things, it was like, 1, no. 2, like even if they did, they were like, they would harvest it and then eat it right now. Like not after it's been sitting in a cooler for 4 hours. But also like, if you're going to do it, like you eat it raw, you can't pull it out and cook it. Like, no, you don't get spirit of the animal. Like if you're going to take it out and like, and put seasoning on it. Like, so no, you can't do that. So we like kept up with this and kept on doing this as we were cleaning a mess of dove. It was like 2 days worth of dove that we're cleaning. And so finally he's like, you know what? Fine. And he just like picks one up and pops it in his mouth and he crunches. And you could hear it as his teeth cut into these valves because you could actually literally hear it crunching through the valves. The muscle and stuff was fine. But you could actually hear him like crunching into the valves and stuff like that as they were trying to break. It was great. Jackass. I maybe feel slightly bad that we convinced him to do it, but also it's a hell of a good story. All right. So all of that is to say that we do have a fibrous skeleton inside the heart. It is not just composed of cardiac muscle. We do have a lot of these connective tissues that my friend was having to crunch through. Okay. The rest of it is muscle, but we do have this sort of fibrous skeleton that everything is going to kind of hold itself around. Now, the cardiac muscles themselves are going to be arranged around this fibrous skeleton, and this is going to help make sure that we change the shape of the heart in a consistent manner. Okay. We're going to have some parts of the heart or some of these bands of tissue be responsible for making sure the heart gets narrowed. It's basically squeezing it in. And then we're going to have another band of muscle that are going to squeeze up. Okay. So we're going to have some muscle that's going to cause the heart to narrow, and then some that are going to cause the heart to shorten. Basically, this means that we're going to be able to squeeze it in multiple directions so that we can push all the fluid out. Imagine like if you were squeezing toothpaste out of a container of toothpaste, or like the toothpaste out of a toothpaste tube. Okay. You can't just squeeze from the side, right? Because some of the fluid or some of that toothpaste is going to get stuck in the bottom. So you have to start your contraction from the bottom. Okay. So what we're going to do here is we're going to have some contraction occur at the top, which is basically then going to fill the ventricles. But then the ventricles, we're going to contract the ventricles starting at the apex of the heart. So down here is where we're going to start our contraction, so that then we can push all the way up from the bottom so that we can squeeze every last drop of toothpaste, i.e. blood, out of our heart every time it beats. This way, none of it gets pulled, none of it gets stuck in the bottom because we're squeezing the middle or anything like that. However, this also means that we have to have a hell of a lot of control over when and how these cardiac muscles go to contract. Okay. We have to control that process very, very, very tightly. And we're going to do that using this special system of signal transduction in the heart that we'll talk a lot more about next time. Okay. All right, we've already talked about some of these blood vessels, so you can see them a little bit more now that we don't have the rest of the heart in the way. So we have our coronary artery here, anterior interventricular artery. We flipped it around the back, you'd see the posterior interventricular artery. Then we have our marginal artery down here. This one that kind of wraps around the left-hand side of the heart, we're going to call that the circumflex artery. So left coronary artery, it splits into our anterior interventricular and then our circumflex artery, where the circumflex is going to kind of move around towards the left-hand side. Then our coronary veins are basically going to drain all of that muscle, and they're going to drain it towards the coronary sinus. Coronary sinus then opens up into our right atrium. Those of you that have known people with various types of heart disorders, whether it's angina or coronary heart disease or a heart attack, here's some kind of little blurbs about these. Your book goes into a little bit more depth into each and every one of these too. I think, yeah, all three of these get their own little blurb in the book. So if you are interested on any of those, make sure you read those. So once again, we have cardiac muscle cells here, and we have our intercalated discs. These intercalated discs are basically going to be made up of these little tight junctions, or sorry, gap junctions and desmosomes. These are essentially ways of us moving nutrients from one cardiac muscle cell to the next. So that say we are propagating an action potential down the membrane of one cell, we do that via sodium and potassium channels opening, right? So changing the charges. Well, the sodium and the potassiums that we're moving around, we can also move through these desmosomes and through these gap junctions so that we can kind of continue propagating that signal from one cell to the next via these intercalated discs in the cardiac muscle cells. Cardiac muscle cells are very much arranged, very much like our skeletal muscle cells were, but instead of it just being basically in a long tube, now we're going to have some tubes that are able to branch out. This is going to allow us to propagate signals in multiple directions in many cases. So we can send one, but now we can kind of offshoot that signal and start it kind of moving off somewhere else. So that we can kind of contract in this big circular pattern rather than just simply down the length of the cell, which is how it worked in the skeletal muscles. All right, so here is our conduction system. The timing wise. Okay, I guess we're good. All right. So say we need to tell our heart to beat. We're going to start telling our heart to beat right here at what we call the SA node. This is the sinoatrial node, where sino is above, atrial tells us we're in the atrium. So this is our sinoatrial node. This is basically a set of cells, or a little cluster of cardiac muscle cells here at the top of your right atrium. These will then, or are going to send out a signal that's going to say, hey, contract. They're going to do this because they leak, but they leak at a very particular rate. Okay. So if y'all remember when we talked about nervous tissue, how we said that we have to reach a threshold potential. And once we send enough signal into that nerve that we hit that threshold potential, then it does the action potential thing, right? So that's when it depolarizes. Well, these don't have just a standard resting membrane potential where they hold the cell at a resting membrane potential. Instead, after they send out an action potential, they're going to sit at the resting membrane potential for just a moment. Okay, so instead of it being flat and then depolarization, repolarization, then back to flat, instead, these are going to slowly change from that resting rate up to a threshold value. Okay, because they have a certain number of leaky sodium potassium pumps that are going to, after we have repolarized, they're going to slowly return or slowly leak up to this threshold value, at which point, when we hit the threshold value, what happens in these excitable cells? They excite, they depolarize, and then go through their repolarization. So they depolarize, then back up, and then leaks, depolarizes, leaks, depolarizes, leaks, depolarizes. And it just does this process over and over again. So these cells do not need a nerve to tell them what to do. Let me repeat that. They do not need a nerve to tell them what to do. They excite themselves. The sinoatrial node will make your heart beat without a brain attached to it. Just beat. Leak, beat. Leak, beat. Okay, and it's going to do this at a rate of around 90 beats a minute. Sinoatrial node. S-I-N-O-A-T-R-I-A-L node. Sinoatrial node. Okay, this is your heart's natural pacemaker. This is what's going to set the rate at which your heart is going to beat. Now, I just said that it sets a rate typically around 90 beats a minute. I am standing up here talking to you all, standing, gesticulating wildly, so much so that I'm sweating a little bit. My heart rate is currently 91, which means my sinoatrial node is able to do this on its own. Y'all's probably nowhere close to that. Our nerves slow our heart down as much as anything. Your vagus nerve, the wanderer, can tell us to slow this down. If our medulla oblongata says, hey, we're just in a resting state, we're just chilling, we don't need to beat 90 rates a minute, it will send nervous impulses out to this to slow down that leaky rate. So that instead of beating at 90, it will instead beat at 70 or 65. When you're asleep, 55. But it's trying to slow it down as much as anything. Now, only under really extreme circumstances, like say, all of a sudden oxygen levels in our tissues get low because of strenuous exercise or something like that, only then will we send additional signal to make it beat faster. Most of the time, we're going to be sending signals out to slow it down. Okay? But that is our pacemaker. It is trying to set the pace that our heart is going to beat. But once again, notice that we don't have a connection between this one and this next spot. Why? Because these are cardiac muscle cells that are able to transduce signals on their own. When one sends, or when one contracts, it will cause the next one beside it to also contract, and so on, and so on, and so on, and so on, and so on. So we can pass that signal from this SA node down to this next set of control cells, just simply through other cardiac muscle cells. Okay. Also keeping in mind that these are not nerves. They are drawn in yellow, so they look like nerves. They are not. These are cardiac muscle cells, not nerves. Can't stress that enough. That is what makes these things cool. Okay. Now, then we get down here to this next set of signals. Okay, so the sinoatrial node, when it sends its signal out, it's basically going to cause all of the atria to contract. So it's going to cause the right and left atria to contract. Well, what's happening then? If they are filled with blood, now they are not. They are going to empty their blood, and they're going to empty their blood down into the ventricles. That's all that we've done now is we've sent blood down into the ventricles. Now what we need to do is we now need to press the ventricles or the blood in the ventricles hard enough that it goes out to the tissues, okay? But once again, we have our toothpaste issue, considering our signal is up here. If we squeeze right here right now, we're going to stick some toothpaste in the bottom of the tube that we can't get out. We don't want that. So how do we get the signal then down to the bottom? And so this is where we get to our next set of our conduction system. We're going to start a second set of signals right down here, what we call the atrioventricular node. That signal has now passed through the atria, and the signal will now reach this set of cells at our atrioventricular node. These cells will then quickly send a signal out through this little band of tissue that we're going to call the atrioventricular bundle, that will then pass into our right and left bundle branches, so splits into right and left sides. And then those go down and they're going to enter what we call the Purkinje fibers, which are going to be down here towards the apex and the base of the heart, okay? These then will end in the actual, in the rest of, or the walls of the right and left ventricles. Once the signal gets out of these specialized sets of cardiac muscle cells, once again, not nerves, then the walls of the ventricles will contract, okay? So basically we get the signal down here, and this signal hits this little bundle, which is basically a whole bunch of excited cells, and they're basically your Paul Revere's of the world saying, hey, get ready, get ready, get ready. And then those signals go out, signals shoot down through these, and then the rest of the walls can contract, okay? This way we give enough of a lag time between the atria contracting and the ventricles contracting to make sure that the ventricles can fill so that they are able to be completely filled with fluid so that we're not pushing air, or at least pushing on a vacuum, but also to make sure that we're able to start the contraction at the base so that we're not going to have to pump blood multiple times because we're getting toothpaste stuck in the bottom of the tube. Does that make sense? Now, this also gives us two places to control heart rhythm. We're going to have nerves talking to our sinoatrial node, telling it to slow down. We're also going to have nerves attached to our atrioventricular bundle, telling it to speed up or slow down based off of our body's needs. Okay, so say we need to tell it to slow down just a little bit, like have a little bit more pause in between our atria and ventricles contracted because we're really filling the ventricles with blood, or that we need to have everything contract really fast because we're just trying to get as much blood out of the heart as possible, okay? We need to control these rates because once again, we can't just live at 90 all the time, or even live at 70 all the time. This also gives us an extra place where we can control these heart rates. However, this also gives us extra places where things can go wrong. If our sinoatrial node goes out and these cells stop working the way they're supposed to, or say you generate a new clump of cells that starts overriding the signals that get sent out by your sinoatrial node, we need to fix that. If you've ever heard of someone having a pacemaker installed, they are literally going to install like a little electronic pacemaker on the outside of their heart, which will then help control or equalize the amount of charges that are going out across the heart because of either combating signals between these two structures, or another structure will sometimes pop up and just kind of make its own set of signals because they're cardiac muscle cells and they're just going to do weird stuff sometimes. Sometimes you'll make a new node, which will start sending out signals in between these two things. So you need to kind of equalize those. That's when someone will have a pacemaker installed. It's basically going to be able to equalize or at least control when signals are going to be going out through these tissues. Once again, though, all of this stuff is controlled by the medulla oblongata. However, it is not initiating the signals. The actual initiation of the heartbeat signals inside the heart are going to come from the sinoatrial node and the atrioventricular node. We can modify it using what's happening in the medulla oblongata, but we can't control it necessarily. Like we can't start it like with a nerve signal. That kind of makes it interesting. So much so that sometimes like when people have open heart surgeries and things like that, and the heart has to be restarted, people would even just like, it was an old way of doing it, but it still technically worked. The surgeon would literally tap the sinoatrial node and it would start the heart beating. So here's some of the ways that we can monitor and control these things, whether it's parasympathetic innervation or sympathetic innervation, depending on what we need. So if we need to speed it up or if we need to slow it down. And that's all because of these special nodal cells that we're going to find in the senoatrial node and the atrioventricular node. Super fun. They're weird. So here we have our leaky channels right here. So we contract and then leak, 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 contract, leak, 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 contract, leak, 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 contract, or leak, 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 action potential, leak, 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 action potential. So we start the signal here in the atria, moves down to the atrioventricular node. So atria contract, loose signal moves down to the atrioventricular node, passes through the atrioventricular bundle, through the right and left bundle branches, out througthe Purkinje fibers. And then once we get to the Purkinje fibers, then we can start to contract. And this way we can start the contraction here at the apex of the heart. So we're able to squeeze the toothpaste from the bottom of the tube. Okay. All right. So that gives us basically, I am now done talking to you for today. So what I want you to do now is get very, very comfortable with looking at the heart. Get comfortable with the idea of the blood flow through the heart and all the structures and stuff that's going to pass through. Maybe even learn some of your coronary arteries and vessels and stuff like that. Get really comfortable with this kind of stuff today. And then when we come back next time, we will do a couple of EKGs. So we will look at an EKG on you. So we actually have a little thing where you can kind of take your own. If you have an Apple Watch, you can just do it that way. So we'll do that. And then we'll also go ahead and dissect a heart. But I want to give y'all as much time as possible to look at these models and stuff like that before we start cutting up hearts. So we'll come back next time, we'll cut up in a heart, and then we'll do some EKGs. And I may make y'all run and then do an EKG again. And we'll talk a little bit more about the excitation of these things and tetany and all that fun stuff next time. So let me know if you have any questions.